Hi everyone, let's begin with the discussion of the virology segment under this rapid revision. Before we start with all the different viruses, it is very important that we cover the general properties, because honestly telling you, if you cover the general properties well, half of your chapter of virology is already completed, because the introduction of every virus will be settled out here. So let's get going, and I'll start with what is the structure of a virus that you need to know. Of course, what you have in the outermost layer is that going to be of the envelope. So what you have at the extreme out is going to be the envelope over here. Can you see that there are some projections coming out of it? Yes. What would you call these? They are coming from the envelope. So can I call them envelope proteins or peplomers? And shortly, I'll tell you one deadly virus which wasted lives in so many ways, that's COVID, and that has these envelope peplomers or these proteins, which I'll be talking about in a while. So there is an envelope and then there are envelope proteins. After that, I can see that inside there is a genome. Either there will be some RNA or there will be some DNA. So there is a genome inside, and that genome is covered by a capsid. Can you all see this covering? That genome is covered by a capsid. So combine karo, you'll say genome, you'll say ma'am either it is ribonucleic acid or it's deoxyribonucleic acid. So some kind of nucleic acid is present and that is covered by a capsid. So dono ko combine karo, something of a nucleic acid and something as a capsid together, can I call it a nucleocapsid? So this and this together is known as a nucleocapsid. What else? So if I look carefully, outermost, what did you have? You had the envelope. Then over here, what do you have? This is the nucleocapsid. Can I say in between the two, I can see one more layer, and that layer is the tegument. So can I say the tegument is present between the envelope as well as the nucleocapsid? Now the structure is completed. However, do you think the same structure will be there in every virus? Not possible, right? So please remember, there are some viruses which will have the envelope and some will not. So most of them are going to contain the envelope. However, which are the ones which are non-enveloped? I have divided them into a list of DNA viruses and RNA viruses. The DNA and RNA viruses that are non-enveloped, and you are going to learn it by the mnemonic PAP and PARCH. What is PAP and PARCH? So remember, PAP is the mnemonic for DNA viruses and PARCH is the mnemonic for RNA viruses. Let's complete them. Right now, you've not done the classification, so you might not know, as to what is the different P's. But abhi ke liye just rat lo and give me 5 more minutes and I'll do the classification also with you. What is PAP and PARCH? PAP is going to be parvovirus, adenovirus and papovavirus. Repeat with me: parvo, papova and adenovirus. These are the three DNA viruses which are going to be non-enveloped. Whereas what is RNA viruses? RNA virus ka mnemonic kya hai? PARCH. What is PARCH? P for picornavirus, A for astrovirus, R for Rio. Rio is the same as rotavirus, C for calcivirus, and hepatitis A and E, basically the ones that cause gastroenteritis. So what is PARCH? Again, there's picornavirus that you had. Apart from that, you also had astrovirus, reovirus, calcivirus, hepatitis A as well as hepatitis E. Remember, PAP and PARCH do not have an envelope. Rest all of them have an envelope. So the envelope story is done. Now let me talk about this story over here. That is the entire nucleocapsid. Do you know the nucleocapsid has a kind of a symmetry? Every genome has a symmetry. Sometimes it can look like this. What do you want to call this? Ma'am, when it's going round and round like this, I'll call it helical. Sometimes you will say it's looking like a little bit of a shape. Kuch shape aa raha hai iske andar. That shape is known as icosahedral. So different shapes and symmetries of that nucleocapsid can be there. Let me tell you, all the DNA viruses are icosahedral. So all the DNA viruses are going to be icosahedral except pox virus. You know, pox is a complex symmetry. You can't comment. It's very complex. So how have I learnt it? Complex. I have learnt it as complex, which will tell me that the complex symmetry is present in the pox virus. Okay, other than that, all the viruses are going to be icosahedral symmetry. Coming to the next one is going to be the helical one. So similarly, all the RNA viruses are going to be helical, and I have learned he is Mr. Fabulous. What is he? He will remind me that I'm talking about helical symmetry, and Mr. Fab is the mnemonic. So let's complete this Mr. Fab. We are going to have myxovirus. Next, R always reminds me of rabies. So we are going to have rhabdovirus. F is going to be filovirus, arenavirus and bunyavirus. Mr. Fab are the viruses which are helical. So remember, he for helical and he is Mr. Fabulous. So simple, quick repeat. All the DNA viruses are icosahedral except pox, because pox is complex. It has a complex symmetry. And who is helical? He is Mr. Fabulous. These are the helical symmetry. So coming back, your entire structure seems to be completed. What can I go on with? I can go on with the classification. How do you classify the viruses? Either they will have a DNA or they will have an RNA virus. So DNA or RNA, both will never be present. Either it's a DNA virus or it's an RNA virus. Now, if you look at the RNA virus list, it is very, very long. Do you want to learn it? Nahin, chota wala list. Ma'am, let's learn the smaller list. So let me make you learn the happy small list, and that is DNA viruses. DNA viruses are said to have a happy small list. So this is the happy mnemonic that you have in front of you. First is going to be H for hepadna. Hepadna reminds me of hepatitis. Which hepatitis? The only hepatitis that is a DNA virus, that is going to be hepatitis B. Now we have another one, the very famous herpes viridae. Herpes viridae, of course, has all the things that sound like herpes. Herpes simplex, herpes zoster. Abhi detail mein we will study. Human herpes virus, Epstein-Barr virus and cytomegalovirus are also belonging to the herpes family. So after that comes the A. A is my favorite, Adenoviridae family. Adenovirus is the virus. Simple. So Adenoviridae, and we have adenovirus. Coming to the next, we have P, and in fact we have 4 P's that we have to go ahead with. So the first one is the pox virus. So remember, pox had smallpox, chickenpox, not chickenpox. That is what I wanted you to note, because many students do this mistake. They say, ma'am, pox, okay, smallpox, chickenpox. No. What is chickenpox? Chickenpox is herpes zoster, so not chickenpox. When I say pox, I mean things like smallpox, cowpox. What are those? Vaccinia and variola are things like smallpox and cowpox. And which pox? Molluscum. Remember, not chickenpox. Coming to the next P is Papoviridae. So now when you look at Papova, all the papo words will come, like polyoma, papilloma, BK virus, JC virus, SV40 virus. I'll be telling you all of these in one line details, okay? So please note papo wale words, papilloma, polyoma, all these short form JC, BK, SV40, all come over here. One more P that we have, simple one, Parvoviridae, parvovirus, easy one. And last one, which you already studied, is a bacteriophage, because I know that bacteriophage, remember what was the shape of a bacteriophage? It was the tadpole shaped structure. And do you remember where in general microbiology have I taught you bacteriophage? I have taught you bacteriophage in the very, very famous, yes, a process that is known as transduction. That is where I am going to teach you this. So I have taught you, in fact, in your general microbiology. So repeating, we have Hepadna viridae, herpes viridae, adenovirus, then you have the famous pox virus, papova virus, papova, polyoma, all of those. Then you have parvovirus, and finally you have bacteriophage. So DNA viruses are happy, and now everything other than this is going to come under RNA viruses. You've done the classification, okay? Now you go on to which of them is big and which of them is small. So which of them is the largest, measuring 300 nanometers, and that is the pox virus family. So remember, pox is the largest, and which is the smallest, and by smallest I mean just 220 nanometers. 20 nanometers means parvovirus. So can I say the size of viruses ranges from 20 nanometers all the way up till 300 nanometers. 20 nanometers kaun sa wala aayega? Ma'am, the tiny little parvovirus is going to be 20 nanometers, and 300 nanometers is going to be the pox virus. This is the size of the virus on the whole. But if I ask you what is the size of the genome, what do you understand by that question, guys? Size of the genome means the size of the DNA, RNA, the nucleocapsid. Uska size kya hai? So who has the largest genome? That is your HIV, that is retrovirus, that has the largest genome. Whereas who has the smallest genome? Best answer will be hepatitis D. If hepatitis D is not given in your options, then you will mark hepatitis B. So especially for FMG students, this came as a question in one of the years, important for NEET and INI also. How it came in the previous years was, They asked you who has the smallest genome, and hepatitis D was not in the options. Then I had to select hepatitis B by default. But if hepatitis D is written in the options, then I will go with D ideally. So please remember, it is hepatitis D followed, if not there, then by hepatitis B. So largest genome is retrovirus and smallest is going to be hepatitis D. Now coming back. So you've done the size of viruses, you've done the size of the genome. Now we can come down to the shape of certain viruses. And I've got a list of these viruses for which you have to know the shape. But how can we learn the shape like this? We should look at diagrams now. Please, did you realize that all the sizes were in nanometers? Nanometer matlab light microscope will not be able to see it. I will have to use an electron microscope to see it. So let's not write it. Let's see it. Directly dekhte hain. Let me begin. Which is the virus? See, black and white photos. Why black and white photos? Because I'm dealing with electron microscopy. It shows you a bullet. Which virus looks like a bullet? So bullet is going to kill. If you shoot someone with a bullet, that person's gonna die. Rabies is also going to make a person die. So bullet kills, rabies kills. Bullet shaped is rabies virus. Coming to the next one, the one that needs no introduction, the one that has wasted everyone's life through and through, and that is the crown one, the one which has these petal-like papillomas. That is of course going to be the COVID or the coronavirus. Coming to the next one, the one that is going to look like a brick. Can you see this is looking like a brick over here? And what is inside the brick? If you look carefully inside the brick, there is a dumbbell that is present. So please note, there is a brick and there is a dumbbell. What is this virus? This is going to be the pox virus. How have I learned it? That instead of brick, can I write box? Yes, I have learned pox is a box. Pox is going to be a box-shaped virus, and the box is very, very heavy. Why is the box heavy? Because the box has a dumbbell inside it. Always learn it like this. Coming to the next one. What is this looking like? A bowl of spaghetti pasta. What is that virus? Ebola virus. Easy way of learning. Ebola is going to look like a bowl of spaghetti or filamentous. Coming to the next one. What is the one that is looking like a round wheel? So I have learnt it as wheel, or you can also learn it as the rotating wheel. You can learn it as the rotating wheel. And why did I say rotating wheel? Because that will remind you of rota. That is rotavirus. In fact, I'll tell you one very nice story for this. That way you will never ever forget. Have you all seen those social advertisements on, especially which are made by agencies outside India? How they depict India. If someone has to make some, organization or some Hollywood or some movie has to be made on India, how do they depict India, which is so wrong and so incorrect? But how do they depict India as a rural country with a lot of slums? That is the kind of depiction they do, and they always show that one scene where there is a kid, a tiny little 5-year-old kid who is running on the roads with a stick and a wheel in his hand. All of us have seen that, and he's rotating the wheel. Bas wo rota hua bachcha. You have to remember that rota hua bachcha. The rota hua, why? Because the rotavirus kid is really gonna cry a lot. Because what does it cause in children? Why am I giving the example of the rota hua bachcha? They will always show you a rota hua bachcha in the slums who is doing diarrhea. A rota hua bachcha in the slums will be shown. He's playing with that wheel and he's doing diarrhea. So diarrhea hoga, bachcha hoga, rota virus hai, and that wheel that he is moving, all that story comes together. That is how you learn the entire rota virus. Okay, coming to the next, my favorite, because it looks like a star. If it looks like a star, I will name it like a star, and that is going to be astrovirus. Coming to the next, the one that looks like a spaceship. Spaceship matlab, it is going to look like as if aliens have come. So alien agar aaya hai, that means it is alien wala. I'm only writing it. Please, this is not written in the textbooks. This is our mnemonic. Alien wala matlab adenovirus. Alien wala is going to be adenovirus. Finally, the last one. Again, books mention it as rod, that okay, it looks like a rod. I have learnt it as cigarette. Why have I learnt it as cigarette? Because that helps me learn the name of the organism. It is a tobacco mosaic virus. So tobacco will obviously remind me of cigarette, right? So tobacco mosaic virus is going to look like a rod or cigarette. If you want, you can quickly revise with me. How was rabies? Rabies was looking like a bullet, ma'am. It's a bullet. Rabies kills, bullet kills. Pox is a box with a dumbbell. It's a brick-shaped virus, and that brick is also going to have a dumbbell inside it. How is Ebola? Ebola is like a bowl of spaghetti. It is like a filamentous organism. Tobacco mosaic virus, exactly like a cigarette, it's like a rod. Coming to adeno was that space vehicle. Space vehicle means alien aega, wo wala. Corona, I don't need to tell you, petal-like papillomas, which everyone knows. Rotavirus, that story of the rotating wheel that everyone knows about. And finally, astro requires no description because it's going to be star-shaped. All your shapes and structures are also done. Now we move on to the rules and regulations that you have to follow in the virology. What are the rules and regulation, and why do I have this Bollywood actress in the picture suddenly? I'll tell you. I will tell you some rules for DNA viruses, then I will tell you their exceptions. Similarly, I will tell you some rules for RNA viruses and I will tell you their exceptions. So DNA se start kare. All the DNA viruses are double-stranded. All the DNA viruses are going to be double-stranded except parvovirus, because parvovirus is the only DNA virus which is single-stranded. Why have I written or why have I shown? Now you know why have I made Aishwarya Rai? Because I want you to remember from the movie Devdas the character of Paro. Do you remember Paro was left alone? In the end, Devdas also died and in the end she did not even have her husband. So Paro was left alone. First and foremost, Paro will remind you that okay, we are talking about parvovirus. Second, you will be reminded that oh, she's crying because she's left alone. She is the single-stranded DNA. She has the smallest group. She's left without anyone. So do you remember? She has the smallest size. Parvovirus is the smallest size, only 20 nanometers. And one more thing that I'm telling you, Paro is crying a lot. As a result, her cheeks have become red. Paro is crying so much that her cheeks have become red, because later on you will study that parvovirus causes a slabbed cheek appearance, which I'm going to teach you in a while. That is the entire story of Paro that you have to know. So repeating, Paro is left alone. So she is single-stranded. Paro is also the smallest. That is why she's crying. She's crying a lot. Her cheeks have become red. So reddish color rash, slabbed cheek appearance is also seen with parvovirus, and DNA virus and exceptions are done. Similarly, you are going to have RNA virus. So pehle to let me tell you the rule. RNA virus ka rule is that there will be a single copy of single-stranded RNA. So one copy, one strand, and it does not have any segments. It is unsegmented. So single copy, single strand, no segments, except is what I want to tell you. So please remember, all of them have one copy. However, retrovirus has two copies. Do you remember the example of retrovirus? HIV doesn't have one copy. HIV has two copies. How will you learn it? From today onwards, you will learn it as Red2 virus. Because retro ki jagah Red2 kar do. Red2 will always remind you that it has two copies of RNA. Usually which RNA is there? Usually single-stranded RNA is there. But ma'am, there is one exception to it, and that exception is the Rio or the rotavirus. What is rotavirus? Let me now tell you. Rotavirus is the double-stranded, the only double-stranded RNA virus. How did I learn it? If you remember, I just told you rotavirus was that rota hua bachcha. Why? Because the rota hua bachcha was having diarrhea. So remember, जो diarrhea वाला है, that is the double-stranded RNA virus. The diarrhea वाला rotavirus, that is the only double-stranded. All the others are single-stranded. Next, all of them are unsegmented. Means if the RNA looks like this, one segment. But I am saying that there is an exception called bira. Everyone knows bira is a very famous so-called alcoholic beverage, which basically means that bira, once someone consumes alcohol, he is going to break off in pieces. He will fall here and there. So now the RNA will not be unsegmented. Now the RNA is going to have certain segments. So basically, if you consume alcohol, you will break into pieces. You will fall here and there. So all of them are unsegmented. But bira, alcohol consume hua to segments hoga. Firstly, what is bira? Bira stands for B for Bunya virus. Then we have I for influenza. Now many students ask me, ma'am, kaun sa wala influenza? Orthomyxoviridae or paramyxoviridae? So remember orthomyxoviridae. That is why many students, instead of learning it as bira, also learn it as bora. So bira, bora, your wish. Now we come to the next one. That is going to be the Rio or the rotavirus. Imagine that rota hua bachcha is also having bira alcohol. And finally we have the very, very famous arena virus. Okay, so now coming back, we have bunya, we have influenza, which is orthomyxoviridae, Rio or rota, and arena. How many segments do they have? Dekho, ma'am, bunya to looks like a 3, B looks like a 3, so that has 3 segments. I can make an 8, so that has 8 segments. Repeating, B looks like a 3, so I am going to make 3 segments. Then I is going to look like an 8, so I am going to make 8 segments. If I combine these two, what is 3 plus 8? 11. So how many segments is Rio virus going to have? 11. And what is 1 plus 1? 2. How many segments is arena virus going to have? 2 segments. So remember, 3 segments, 8 segments, add kara, so Rio virus has 11 segments, and add kara, that has arena virus 2 segments. So if you have beta alcohol, you will break into segments. Otherwise, all the other viruses are unsegmented. So repeating, all of them have 1 copy, but red 2 virus has 2 copies. All of them are single-stranded, but diarrhea wala is double-stranded. So Rio rotavirus is double-stranded. All of them are unsegmented, but beta virus is going to be segmented. I hope that is going to be okay with everyone, because now we can move forward and talk about how are you going to grow the viruses. That is also important. Finish it off over here only. How are you planning to grow the viruses? So please remember, in virology, you don't have the blood culture, chocolate agar, blood agar, nutrient agar. These agars are not there, because viruses you don't grow in culture media. Viruses either you can grow in an animal, number one, so put it into an animal, or you can grow it in an embryonated egg, because of some animal or of human, some embryonated egg, or you can grow it in cell lines. What is common to all? That ma'am, you basically, virus requires some kind of cellular support. You can't just grow it with the help of blood or chocolate or egg or serum like you were doing for bacteria. Viruses don't need just food, they need proper cells. Either you give them the cells in the form of an animal or an egg, or you give them some cell lines in the lab. So cell chahiye, that is when a virus will grow. So let me take the example of animal number 1. So for example, if you inject something in the brain of a suckling mice, let me take an example. Coxsackie A and B were injected in the brain of a suckling mice. How will I get to know whether it is Coxsackie A or Coxsackie B? So please remember, Coxsackie A is going to cause flaccid paralysis, and Coxsackie B is going to cause spastic paralysis. And how did I learn which is flaccid and which is spastic? I simply know, go alphabetically. A comes before B, तो पहले A आता है, फिर B आता है. Similarly, F comes first and S comes later. So simple, A comes first, B comes later, F comes first, S comes later. So that is how you arrange them in alphabetical order. That is how you grow it in animals. How do I grow it in embryonated egg? Firstly, this is how an embryonated egg looks like. क्या parts होते हैं? Ma'am, there is a chorioallantoic membrane, that is CAM. Then I can see there is a yolk sac that is present over here. You can grow it in the yolk sac. Then there is something known as an amniotic cavity. Then there is something known as an allantoic cavity. 4 things are there: chorioamniotic membrane or chorioallantoic membrane, yolk sac, amniotic cavity, allantoic cavity. So what do you grow, and what are these pictures shown? I have shown one picture of a car. Number 1, car ka koi photo hai, and some best friend, yaar, yari, that kind of picture is there. So let me repeat it. If I ask you, what do you grow in a yolk sac? So you will say that, ma'am, there are boys, they are roaming around in a car. Car mein roam karte karte, when they are roaming around, they are roaming around in a luxurious car. So what is their usual swag going to be? They are going to be saying yo-yo. So you are having yo-yo for yolk sac. They are roaming around in a luxurious car. And what is car? Remember, A for which virus? Arbovirus, and C and R for 2 bacteria which are grown in the yolk sac, chlamydia and rickettsia. Remember, so yolk sac, yo-yo, car, matlab it is going to be the chlamydia, rickettsia and arbovirus. In case you forget in the exam, A se kya tha? A se arbo tha, adeno tha. So remember, it is A car. That will give you a hint. It is arbovirus that you have to go in for. So yo-yo kara, car mein. Okay, now we say that, okay, what is in the chorioallantoic membrane? CAM mein. So we have VVHH. What is VVHH? We have vaccinia, variola, HSV1 and HSV2. So like I told you, in the chorioallantoic membrane, we have the VVHH, vaccinia, variola, HSV1, HSV2. Now look at these two. Look at the amniotic membrane and the allantoic membrane. So you will say, ma'am, allantoic mein to, if you look at allantoic cavity, you have grown, number 1, yellow fever. Secondly, you have grown, or there has been a vaccine preparation of influenza. There has been a vaccine preparation of influenza, and there has also been growth of rabies. So growth of rabies virus is also done. So please remember that all of the boys, number one, boys se related hai. All of the boys do what? If you put them in a car, they will do yo-yo. If you put them together, if all of them have come together, you will say, oh, they become best friends, ma'am. Boys become friends instantly. They just need to shake hands and they are friends after that. Best friends for life. So all of them together become yari. Means yellow fever, rabies virus or yellow virus, rabies virus and influenza ka vaccine preparation, not influenza ka growth. Influenza ka growth happens in the amniotic cavity. So please note this difference. That is why I'm saying boys are very protective about their yari. You can't say anything about a boy gang to anyone, isn't it? So boys are very protective about their yari will give you a hint that you are making the protection for influenza over here. You are making the protection for influenza or the vaccine for influenza over here. But in amniotic cavity, you are growing the influenza virus. So coming to the summary. Chorioallantoic membrane, vaccinia, variola, HSV1, HSV2. Yolk sac, ma'am, yo-yo kara, car, chlamydia, arbovirus, rickettsia. Amniotic cavity में influenza virus is grown, but allantoic cavity में yellow fever virus, rabies virus and influenza vaccine is grown. And that is the summary of the entire egg. One very difficult topic that you have. I hope we have sorted it. So animal growth done, embryonic or embryonated egg growth done. But actually what is practical is that in the lab we use cell lines. How many types of cell lines do we have? Primary, secondary and continuous. Means primary will allow cell matlab division. Primary will allow cell to divide for approximately 5 to 10 times. Secondary will allow a little more, 10 to 50 times. Continuous will allow unlimited. The name only tells you, continuous divisions will happen. Very difficult to learn, ma'am. How should I learn which cell line is coming under primary, secondary, continuous? This is not important for FMG students, but this table is important for NEET and INI-CT. So please note, for primary cell lines, you have the kidney of the monkey, which is used as a cell, or you can use the amniotic from a human, or you can use the embryo of a chick. So you realize कि मैम primary cell line में ना सब कुछ primitive use हुआ है, like the amniotic which is from the fetus in the humans or the embryo of a chick. So everything primitive, fetus, embryonic, that has been used. And rhesus kidney, rhesus तो मतलब monkey तो anyway they are primates, monkeys are our ancestors, so they are also very primitive. Anything primitive mentioned will come under primary cell line. But if I mention human fibroblast cell line, इसमें कोई fetus, embryo ऐसा कोई word नहीं है, that becomes a secondary cell line. And that is important because human fibroblast cell line has been used for the growth of cytomegalovirus, a very famous and an expected question. So repeating once again, anything that is to do with amniotic, embryo, fetus or monkeys, all primary. Human fibroblast cell line with this M and W, MRC5 and WI38, these are secondary. And after that, no matter what is written, HeLa, Hep2, KB, McCoy, Vero, Detroit, BHK, Chang, CILK, all of these come under continuous. I don't have to learn that list. Primitive चीजों का नाम याद करो. M, W and anything with related to human fibroblast is secondary and everything else becomes continuous. We are done with the cell lines. These are the three ways by which any kind of virus can be grown. The last part of general virology that you have to know is what are the different inclusion bodies that you have. So quickly, which are the ones in the cytoplasm, and then you'll tell me about which are the ones that are going to be present in the nucleus. So let's get going. In the cytoplasm, Negri bodies, let me show you a picture. You can see that, yeah, this must be a cell and these are the Negri bodies. Do I need to ask you this, that Negri bodies are seen in actually which kind of virus? They are seen in rabies, but in which part of the brain are they seen? So definitely they are seen in rabies, but they are seen in the hippocampus and the cerebellum. These are the two places where they are noted, in the hippocampus and the cerebellum. Coming to the Pasteur and Guarnieri bodies, all time confusion. Some books say variola, vaccinia. Kya confusion hai? So please remember, Guarnieri, or if I write vaccinia, vaccinia is going to have lots of C's which I can easily convert into G's. So remember, Guarnieri bodies are seen in vaccinia and then Pasteur bodies become for variola. Coming to Bollinger bodies, very easy to learn. Whenever you put a ball, कोई football, basketball, कोई भी game होता है उसमें क्या होता है? Foul. So Bollinger bodies are seen in fowl pox. Bollinger bodies, always remember, कोई भी football होगा तो उसमें foul होगा. Bollinger bodies are seen in foul pox. Molluscum contagiosum shows you what bodies. Molluscum contagiosum, let me show this to you separately. This is a case of molluscum contagiosum and I am integrating this with dermatology also. Molluscum contagiosum usually presents with umbilicated lesions. Molluscum contagiosum usually presents with umbilicated lesions. Umbilicated matlab there is a depression in the center. Depression. Why? Because if you press it, some curdy material is going to be extruded. There is some curdy material that is going to come out. So, chota sa depression hota hai. If you press it, some curdy material will come out. How does it look like under the microscope? You can very well see it is a cup-shaped lesion. It is a cup-shaped lesion, and if you go deep into the cup and you zoom in into the cup, you will realize that there are some round bodies. What are these guys? These are the famous bodies of molluscum contagiosum, and these bodies are classically known as the HP bodies. So let's know the full form. What is HP? HP stands for Henderson-Patterson bodies. HP is going to stand for Henderson-Patterson bodies, and please note, now again we write it as HP, but I want to tell all the students preparing for INI CET that Henderson-Patterson bodies are going to be positive for a stain called the floxine tartarate stain. Very important question that you get in the exam. Floxine tartarate stain is going to be positive in these Henderson-Patterson bodies. So coming into this, I hope everyone is settled. Umbilicated lesions, material will come out microscopically cup-shaped with Henderson-Patterson bodies, and P for Patterson, P for floxine tartarate stain, that is going to be positive. So coming back, what does molluscum show you? Molluscum tends to show you HP bodies. All of these were in the cytoplasm, because now I will ask you which are the bodies which you see in the nucleus. Whenever they are in the nucleus, you call them Cowdry type A, Cowdry type B, and there's a very famous mnemonic for this that you study, that is Hey Bap. What is Hey Bap? So firstly, of course, pick up Cowdry type A and Cowdry type B. Okay? So Cowdry type A are going to be seen in herpes and yellow fever. In herpes and in yellow fever. So remember, yellow fever में कौन सा bodies? Tories bodies. Remember how I'm writing it? Yellow fever में we are going to have Tories bodies. Okay, yellow fever done. What is herpes? Herpes में usually lips पे lesion होता है ना. You must have heard of herpes labialis. तो herpes में lips पे lesion होता है. That is why I have learnt it as lips. Leib-Schultz bodies are noted. Simple. So remember, herpes is going to show you Leib-Schultz bodies, and yellow fever is going to show you Tories bodies. Coming to BAP, that is who has Cowdry type B. Cowdry type B are seen in adenovirus and poliovirus. Adenovirus and poliovirus show you Cowdry type B. All of these are going to be in the nucleus. I hope that is okay with everyone, because now I will ask you, both intranuclear and intracytoplasmic inclusion bodies are seen in. So remember, there are two organisms. One is going to be measles and the other is cytomegalovirus. Let me show you their pictures. I think cytomegalovirus to everyone knows. Cytomegalovirus shows you the very famous owl-eye bodies. So if you go into it, you will realize that yes, there are intranuclear inclusions also. That is what gives it owl-eye. And there is a bunch of intracytoplasmic inclusions also. Both of these are noted in the same owl-eye. So cytomegalovirus, intranuclear plus intracytoplasmic. What is measles? Come on, measles mein you know one giant cell from pathology. Measles mein kaun sa wala giant cell? We have learnt it is the Warthin-Finkelday giant cells. And yes, Warthin-Finkelday giant cells, if you look into this giant cell, you will again visualize there are lots of dots, lots of pink color dots that are present in the nucleus and in the cytoplasm. So it's going to show you intranuclear plus intracytoplasmic inclusion. So repeating once again, majority of the list that I taught you, all of it was intracytoplasmic. Then I taught you a mnemonic called Hey Bap. Hey Bap is going to be intranuclear, and then two of them which are going to show you both intracytoplasmic and intranuclear are cytomegalovirus as well as measles. Now with that, you finish off. You might feel that you have done some 30 odd slides of virology, but trust me, you have done half of virology, because this is where you get maximum questions, and this is what sorts all the difficult part of virology for you. Hi everyone, let's begin with the discussion of the DNA viruses, and I just now taught you the different DNA viruses come under a common mnemonic, that is the happy mnemonic, right? So let us begin with the largest family that I see over here, which is the herpes viridae family. And before I start, let me tell you what all viruses come under it. So first and foremost, we divide it from HHV1 to HHV8, human herpes virus 1 to 8. Out of them, 1, 2 and 3 are a group. Why are they a group together? Because all of you have heard of herpes simplex 1, herpes simplex 2. So herpes simplex 1 and 2 is human herpes virus 1 and 2. Simple. 1 and 2 over here is 1 and 2 over here. What is human herpes virus 3? That is the zoster virus. I hope you remember zoster virus is something that causes chickenpox and zoster itself. Zoster also known as shingles, right? So yes, that is what is human herpes virus 3. Now let's come to the remaining 6, 7 and 8. Human herpes virus 6, 7 and 8 are not given any other names. They are known as human herpes 6, 7, 8, right? So the only different names that now I am left with are 4 and 5. Remember, human herpes virus 4 is known as Epstein-Barr virus and human herpes virus 5 is known as cytomegalovirus, and that wraps up all of your classification. So exactly the same order is what I'm going to take up for this. I'll start with HHV 1 and 2. 1 and 2, as I told you, is nothing but simplex 1 and 2. And let's try and do a difference between HSV 1 and HSV 2. I'll tell you the clinical way of picking it up. That will give you a lot of clarity. Please note that whenever you are taking up any lesion above the waist, so whatever lesions are going to be above the waist, say they talk about the lips or the face or the gingiva or anything above the waist, that is going to be HSV1. That also includes all the orofacial lesions. That also includes all encephalitis, meningitis, because brain is also above the waist, right? Hopefully in most of us. Okay, so having said that, HSV1 is everything above the face. This means the face, the oral cavity. So mucosa se aayega. What is the transmission going to be? The transmission is going to be via the mucosa. Which ganglion is it going to stain? Something which is above the waist again, and that is going to be the trigeminal ganglia. So everything for HSV1 can be learned in a simple mnemonic, that is above the waist, whereas everything for HSV2 will be learnt as below the waist. And what do I understand by below the waist? So all the sexual related or the genital lesions that are going to be present, they are all the genital ulcers, they are below the waist, so they will come in HSV2. That means that even neonatal herpes will come in HSV2. Why? Because neonatal means that there is going to be mother to child. Mother to child through vaginal canal is of course going to be via below the waist only, right? So that is one route. Or genital lesions are of course going to occur via sexual. So what do you think is the ganglia in that region? Ma'am, sexual, genital, pelvis, sacrum. Sacrum is over there. So what ganglia is going to be the one in which the virus is going to sit? It is the sacral ganglia. That wraps up all of your table. Now most of it is settled for us, but some things I want to tell you in a little bit more detail. So for example, for HSV1, you asked me which is the most common site. Everything was above the waist now, on the oral and the facial region. So the most common site is the buccal mucosa. The buccal mucosa inner aspect, that is the most common site. And what lesion is going to occur most commonly over there? The gingivostomatitis. There is involvement of the gums. So buccal mucosa and gingivostomatitis most common. But what is the lesion that occurs again and again in terms of recurrence? Which is the most common recurrent lesion? And that is labialis. Herpes labialis is going to occur again and again. So please note, read carefully. If they ask you the first time lesion is the gums, gingivostomatitis. But if they ask you the recurrent lesion, that is going to be herpes labialis. What else? I just taught you that CNS infections occurs. Can you remember encephalitis, meningitis above the waist? So where do you think and which virus do you think is more common? HSV1 is going to be much more common than HSV2 because it is above the waist. In fact, do you know, for encephalitis, this is said to be the most common cause of acute sporadic viral encephalitis. That is viral encephalitis, sporadic variety. This virus, HSV1, is the most common. What else? Which part of the brain is affected? Encephalitis mein, it is the temporal lobe affected. Similarly, we have meningitis. Meningitis is also going to be above the waist, so of course it is HSV1. However, please remember, so I'll write HSV1 is more common than HSV2 over here. However, there is something referred to as mollaret's meningitis. Now, what is mollaret's meningitis? You can see in mollaret, there are a lot of L's that are present. Mollaret's meningitis. So, this is a meningitis having lots of lymphocytes. It is a recurrent lymphocytic meningitis. It is a recurrent lymphocytic meningitis, and this is more common actually with HSV2. So, please note, all the meningitis encephalitis are more common with HSV1, but mollaret's meningitis is more common with HSV2. Okay, let's move forward. Can this happen apart from these patients? Can it happen in healthcare workers? Of course. Please note that this is also seen in healthcare workers. Can you see there is a small lesion that has developed over here? This can occur in healthcare workers, and that is known as herpetic whitlow. So I have learned whitlow is going to occur in workers, healthcare workers. So how? Because see, for example, dentists are anyway dealing with the oral cavity only. So if some patient has a lesion in the oral cavity and the dentist is working, so on the fingers, on the hands, exposure can occur. So this can occur in healthcare workers. Workers matlab there is going to be whitlow. Other than that, it can also occur in gladiatorum. This is anyway sounding like a gladiator. It is sounding like a wrestler. So yeah, this is known as the wrestler's herpes. And other lesions with herpes also known as eczema herpeticum and erythema multiforme, if you want to remember. But what you have to remember is the fancy names. That is gladiatorum for gladiator and wrestlers, known as wrestler's herpes, and whitlow for workers. So that is for healthcare workers. Now, before I proceed, going back to the table, do you remember for HSV2, I told you genital lesions honge? And if I'm saying that there are genital lesions, then of course, by default, I mean to say there is a genital ulcer. So I want all of you to revise this point with me, that when we were talking about genital ulcers, we always used to do a classification. And that classification was whether the genital ulcer was painful or whether it was painless. Now, when I'm talking about a painful genital ulcer, I think in the bacteria, you remember Haemophilus ducreyi, because we had learned crying with pain. And now I will write HSV2, herpes simplex virus 2, that also causes a painful genital lesion, versus what was in painless. So we had learned syphilis. That is how we learned it, right? That syphilis is everything going to be painless. There is a painless ulcer also, and there is going to be a painless lymph node also. Everything is painless, syphilis. Other than that, we had learned something called lymphogranuloma venereum. See, the ulcer is definitely painless. That is why I've put it under this category. But L for L, the lymph node is going to be painful, and that painful lymph node was referred to as buboes. Finally, we have Klebsiella granulomatis, which causes donovenosis. So Klebsiella granulomatis also causes this particular infection, causes painless genital ulcer. So remember, syphilis is painless, lymphogranuloma venereum is painless, but the lymph node becomes painful, and Klebsiella granulomatis is also painless. Painful mein kya aata hai? There is crying with pain, that is Haemophilus ducreyi, cry, ducreyi for cry, and finally HSV2, and that is how you are going to learn this. So I hope everyone's okay with this information and we can move forward. So this is how we had learned about the genital ulcer. Let's quickly go on to the lab diagnosis. What kind of a smear do I take for the lab diagnosis? We are going to take the Zank smear, and if I ask you what are the bodies that you're going to see, so you will ask me that, ma'am, this I know, there are some bodies that you see in herpes, and I always told you that herpes में एक lesion होता है, that is herpes labialis. Lips are very commonly involved. So the bodies are also referred to as the Lieb-Schultz bodies. I gave you this mnemonic in the general virology, isn't it? Other than that, please remember the Zank smear for herpes shows you 3 M's. You know the picture or you don't is irrespective, but you need to know these 3 M's. What are the 3 M's that are a must to know? Number 1, you are going to have multinucleation. You will see multinucleated giant cells, so multinucleation. Second, the nuclei are going to be molded. There is molding of the nuclei. And third, there is margination of the nuclei. This is a ratta that you will have to do. These are the features of the nuclei stimuli that you notice. Number one, they are multiple, multinucleated, they are molded, they are marginated. So please remember, if we say 3Ms and we say the Leib-Schultz bodies, they are definitely talking about herpes. And what is the smear that you make for the herpes? You are going to make a Zank smear. Zank smear question has come a lot in NEET and FMG exams. You have to know this. What is the treatment, guys? The treatment is going to be acyclovir, and if there is resistance, then foscarnet can be used. But yeah, the primary treatment is going to be acyclovir. In case of resistance, foscarnet. That wraps up HSV 1 and 2. So human herpes virus 1 and 2 is done. Now we go to human herpes virus 3, and human herpes virus 3 is the zoster or the varicella zoster. What does it cause in children? In children, it causes chickenpox. And what does it cause in adults? In adults, it causes herpes zoster or shingles. So let me quickly make you go through chickenpox. Chickenpox, you know, is very important in pediatrics, but quickly let us settle it over here as well. The question that came in the exam recently was the incubation period. What is the incubation period of chickenpox? Roughly 14 to 21 days. So this is a question in the recent INICET that was conducted in 2023 May. So please note, they asked you what is the incubation period, and that is 2 to 3 weeks, 14 to 21 days. Then I have further learned, all of us know there is a rash in chickenpox, and that rash is known as what? It looks as if there is a dewdrop. So number one, it is a dewdrop, and or around it, so you can see the dewdrop, and around it there is a reddish color base, erythematous base. So that is looking like a rose petal. This is known as a dewdrop on rose petal. So can you all see the dewdrop and then red color rose petal? That is the description of chickenpox rash. But I have learned CP mein CP. What is CP mein CP? That means that in chickenpox CP, the rash will start CP, centripetal. So they will ask you that the rash that you have will occur where? Centripetal. Centripetal means on the trunk. It will first start on the trunk and then it will go to centrifugal spread. Centrifugal means it is going to go out. So for example, if this is the body, it is first going to start on the trunk in this region, and then it's going to go out and it's going to spread in centrifugal direction, and centrifugal spread will occur on the fugal flexor surfaces pe hoga. So please note, they will always ask you where does it start and where does it spread. So remember, starting is CP mein CP, chickenpox is centripetal start. Okay, why am I telling you this? Because the opposite of this is going to occur in smallpox. The opposite of this is going to occur in smallpox, and that you would know that if they ask you, okay, where does the rash start, then you will say, acha, it will start centrifugal, it will start over here and it will come centripetal, then it will come towards the center. So now you shouldn't get confused, na? Chickenpox mein CP mnemonic will tell you chickenpox may start yahan hoga and then it's gonna go to the centrifugal direction. Now let's move forward. Imagine you have a case of chickenpox. Chickenpox is a virus, herpes zoster virus. But what are the complications that a person can have? Imagine virus ke upar bacteria infection ho jaye. So that is going to be secondary bacterial infections that can occur. That is most common. So anyway, the person had a virus, skin lesion, skin rashes with virus. On top of that, the person ends up having bacterial skin infection. That is going to make it very complicated. So skin ka problem. But if I ask you which is the most common extracutaneous complication, then you're going to say there will be CNS involvement, brain involvement. That is what makes it very complicated. And what brain involvement is known in chickenpox? You know this question from medicine. There is benign cerebellar ataxia. This is the brain involvement known in chickenpox. But can chickenpox affect pregnant mothers? So many times you heard that there is a female who's ended up having chickenpox in her pregnancy. That can become the most serious complication because varicella pneumonia is usually seen in pregnant women. So firstly, it is a risk to the mother also because varicella pneumonia can affect her. But can it go from mother to child? Yes, it can affect the child as well. So please note, when it affects the child, it is known as the fetal congenital varicella syndrome. That is when infection, congenital, when it has occurred within 20 weeks of intrauterine life. So what happens when from mother, there is a child that is going to get affected, mother to child. Everything is going to become chota chota. So remember, in varicella syndrome, that is how we will learn it. In varicella syndrome, we will learn everything is going to become very, very small. Very small means, now start reading. What is the size of the skull? Microcephaly. What happens to the brain? Atrophy, cortical atrophy. What happens to the limbs? You can see that there is the hypoplasia. No development has occurred. There is limb hypoplasia. And look at the skin. If you see the skin, it is contracted. See, skin is also getting chota. It is getting contracted. So that is known as cicatricial skin lesion. So repeating, in varicella syndrome, everything is becoming very, very small. Okay, very, very small. What is that? Microcephaly, cortical atrophy, limb hypoplasia, cicatricial skin lesions. And that definitely wraps up the childhood problem of chickenpox. But what about herpes zoster, which is seen in adults? So please note that herpes zoster or shingles is nothing but the same virus. It's the same herpes zoster virus, but this got infected during childhood. But this person, after that chickenpox to theek ho gaya, but the virus is remaining latent in some. Its virus stayed back, and virus has gone and hidden somewhere in the body. It has remained hidden in some ganglia, and now it is going to get reactivated. It may be in some time in adults. So when would it get reactivated? Either in some very, very old age, number one, some 50, 60, 65-year-old person, or whenever the immunity of the body goes down. Patient says, I was under prolonged fasting, or HIV patients, or maybe some organ transplant or transfusion. So some kind of immunocompromised state hoga. So that sleeping sleeping virus will suddenly wake up, right? That is what results in herpes zoster. So how is this different from chickenpox? If you remember, in chickenpox, the first thing that I told you was the rash was everywhere, centripetal everywhere. But in your herpes zoster, you can see it is a unilateral dermatomal distribution. There is a unilateral rash which is going to be along a segment. Which segment? Along, according to the dermatomes, most commonly D3 to L2 dermatome is affected. D3 to L2 dermatome is affected, and one very important point of this is that this is a painful rash. Pain is a feature of herpes zoster that you have to consider. So it's going to occur in this one. Hamesha unilateral yaad rakhna. This is a big hint, and along some segment, along some dermatome, it is going to happen. So now you say that, okay, acha ye to main presentation ho gaya. How can the patient complicate? See, firstly, can you see the involvement of the rash? As I told you, the rash is going to be unilateral. So you can very well see there is a unilateral rash that we have over here. And other than that, I can see the involvement of the eyes that has occurred. Yes, zoster ophthalmicus is definitely one of the complications that can occur. So eyes may, there are ulcers and there are rashes around it. So this is one complication. But what is the most common complication? The most common complication is nerve pain. Post-herpetic neuralgia is the most common complication. Neuralgia ka matlab everyone knows, nerve pain. So wherever, for example, if over here the rash had occurred, patient will complain of immense pain in this region. So post-herpetic neuralgia is the most common complication that you have to know. However, you must have heard of many celebrities also get this. And what is this complication known as? Ramsay-Hunt syndrome. What is Ramsay-Hunt syndrome? Firstly, in Ramsay-Hunt syndrome, which nerve and which ganglion is getting affected? It is the geniculate ganglion of the seventh nerve. So, kaun se nerve ka palsy hoga? Facial nerve, seventh nerve ka palsy hoga. So, what all is going to get affected in Ramsay-Hunt syndrome? Of course, ma'am, if the seventh nerve is affected, facial nerve palsy is what I'm expecting. Second, so this is a facial nerve palsy. You can see the deviation of the face. Other than that, what else can you see? I can see some ear involvement. There are vesicles in the external auditory meatus along with ear pain, otalgia. And then I can see a problem in the tongue, or I can see a loss of taste in the anterior 2/3 of the tongue. So repeating once again, whenever you're dealing with the case of Ramsay-Hunt syndrome, number one, there will be a problem in the facial nerve. Maybe there's a deviation of the face. Then you can see some external auditory meatus having some vesicles, some earache, and you can see loss of sensation in the anterior 2/3 of the tongue as well. That is Ramsay-Hunt syndrome. Okay, this is settled for us. So what all are we done with? We are done with HHV1, HHV2 as well as HHV3. Have a look at this table. Now I'm going to go forward and talk about HHV4, and that is Epstein-Barr virus. So now starting with human herpes virus 4, and that is the Epstein-Barr virus. The first thing that you have to know is the pathogenesis, because you get a lot of questions on these. So for example, if this is the Epstein-Barr virus, the first question that will be asked is how does it enter the body and where does it attack? So EBV has a B in it. That means Epstein-Barr virus is going to go and enter the B cells of the body. Question 1. How does it enter the B cell of the body? It is going to go and enter via a receptor called CD21 or CR2. Again, a previous year question. Once it has entered, what are the genes that Epstein-Barr virus is going to have? It has LMP1, it has EBNA2, and it has interleukin 10. The virus has interleukin 10. So what does it have? It has 1, 2, 10. It has 1, 2, 10, means it has LMP1, it has EBNA2, and it has interleukin 10. Go back to your pathology lectures, because you would know that interleukin 10 is pro-inflammatory or anti-inflammatory. It is AN10, that is how we had learnt it. Interleukin 10 is anti-inflammatory. It will not let inflammation happen in your body, means it's not letting you react, it's not letting you fight. Other than that, LMP1 and EBNA2 are responsible for the diseases. Now, what are the diseases that I'm talking about? Before I proceed, question 1, which cell of the body? B cell. Question 2, what is the receptor? It is CD21CR2. Then we have what is the LMP1, EBNA2 and interleukin 10. So these are the 3 things that it releases. Please note that this EBNA2 is responsible for activating an SRC proto-oncogene. This information is only important for INI CET aspirants. It acts on SRC proto-oncogene. So they'll ask you a simple question. SRC proto-oncogene is implicated in the pathogenesis of which virus? And that is Epstein-Barr virus. Now coming to what are the clinical features that it has. Please note, the most common disease which you know of with EBV is infectious mononucleosis. What are the other names for it? Number 1, it is known as kissing disease. Why kissing disease? Because it spreads by very close contact. And the other name for this disease is known as glandular fever. Now, the purpose of calling it glandular fever is that number 1, the patients are going to have fever, that is obvious. Glandular means that lymph nodes, salivary gland, jitna bhi glands hai, maybe liver and spleen, these are all enlarged. Glands become big, and that is why you call it glandular fever. That is number 1. So, you know, this infectious mononucleosis actually shows you something under the microscope. Please note, microscopically, do we all know this very famous girl and the ballet skirt appearance? There are cells which are showing you the ballet skirt appearance, and these are referred to as the Downey cells. So, if you are asked that what are Downey cells, you will say Downey cells are nothing but the lymphocytes only, but those lymphocytes have become little ajib. Means when I draw a lymphocyte, I always draw a round cell. Suddenly, I'm drawing a cell which is looking like a ballet skirt. So that is what I called it. I called it the very, very famous ballerina skirt appearance. Remember, ballerina skirt appearance or Downey cell aaya, you will mark infectious mononucleosis. But now comes the trick question. What kind of a lymphocyte is it? Remember, it is a CD8 T lymphocyte, and don't get confused. If I ask you Epstein-Barr virus enters into which cell of the body, it enters into which lymphocyte? B lymphocyte. But imagine B lymphocyte to hijack ho gaya, right? B lymphocyte has been hijacked. So which is the cell who's going to come and save the hijacked cell? So there comes a ballet skirt who's going to come and save it. That okay, I'm your cousin brother, I will come and save you. So the T lymphocyte, the CD8 T lymphocyte comes and says, I will rescue my brother B cell. So if someone asks you what is the composition of the ballerina skirt appearance, that is a CD8 T lymphocyte. What else does it cause? We've only spoken about infectious mononucleosis. Other than that, it also causes malignancies. Under malignancies, it can cause every kind of malignancy that you've heard of. It can cause a carcinoma. It can also cause a sarcoma, which many people are not aware about. And it can then further cause lymphomas also. So please note, so the most common carcinoma that it causes is the gastric carcinoma, right? So gastric CA is the most common. Other than gastric carcinoma, it also causes nasopharyngeal carcinoma. So if the most common carcinoma is asked, you mark gastric carcinoma. Other one is NPC, nasopharyngeal. It can also cause a sarcoma, which many students are not aware of, and that is a leiomyosarcoma. So leiomyosarcoma is something that you have to definitely consider. Now comes lymphomas. Can it cause Hodgkin's lymphoma? Yes. Can it also cause non-Hodgkin's lymphoma? Yes. What all comes under non-Hodgkin's lymphoma? It can cause Burkitt's lymphoma. It can cause diffuse large B-cell lymphoma. It can cause post-transplant lymphomas, and the list is possibly endless. There are so many lymphomas that this can come up with. So remember, carcinoma, sarcoma, lymphomas, all are known with Epstein-Barr virus. But forget malignancies. Other than malignant conditions, does it also cause oral hairy leukoplakia? Yes. So please note, this is famous for causing oral hairy leukoplakia, which is coming under the miscellaneous variety. So there you go. And if you're targeting the INI-CET exam, let me tell you, you would also want to know about Duncan syndrome. Again, I'm telling you something which is important for INI-CET aspirant. Duncan syndrome is a lymphoproliferative syndrome that is there, which is also going to have you have association with EBV. So that wraps up all of this. How am I going to do the diagnosis? I think for the diagnosis of EBV, I have already told you the microscopic examination showing you the very famous ballerina skirt appearance, that is the Downey cells, that everyone knows. But what is the question that you get most commonly for antibody detection? And that is going to be heterophile antibody detection. I think you've heard of these two tests. There is something known as a Paul Bunnell test, and there is, which is an old version of it. Nowadays, what is done is a new test, and that is a monospot test. Monospot test is the new version of the test, and that is what you have. That is heterophile antibody testing. But remember, let me tell you, these are still not considered very specific. What are specific antibody testings? These are the three that you have to know. Did I tell you something called LMP1, EBNA2? These were parts of EBV. So antibodies against EBNA, antibodies against early antigen, which will allow early diagnosis, antibodies against viral capsid antigen. So these are all virus parts, and if these are specific to the virus, so antibodies against these parts will make a specific diagnosis. But if they ask you which is the most sensitive and specific method overall, not the antibody detection, but overall which is the most sensitive and specific method, I think that is understood that genetics ke upar to kuch nahi hai, right? So nucleic acid hybridization testing becomes the most specific and sensitive method. However, if they ask you what are the specific antibody testings, then you have early antigen, EBNA, and viral capsid antigen. Finally, talking about the treatment aspect, for oral hairy leukoplakia, there is definitely treatment of acyclovir that has been considered, and rituximab is also being considered for the treatment of EBV. So that wraps up your human herpes virus 4 all in all. Means we are going to go on to what? We are going to go on to human herpes virus 5, and that is going to be cytomegalovirus. Did you ever wonder why did you call it megalovirus? Because the largest member of this family is cytomegalovirus. It's not the largest virus overall. I hope you remember the largest virus overall was the pox virus, right? That is different. I'm saying of this family, which is the largest virus, the megavirus, cytomegalovirus. And we are the only reservoirs. For this, I'm going to show you a picture also whenever cytomegalovirus comes. So cytomegalo means he has put out his arms. As soon as I say the action of putting out the arms, we all think of the very famous SRK, that is Shah Rukh Khan. So please remember, where is it multiplying? You say cytomegalo, ma'am, it's so mega. How is it? Who is it working like? It is working like Shah Rukh Khan. Means it is multiplying in the salivary gland, the respiratory tract, and the kidney. Remember, it is working as Shah Rukh Khan. That is salivary gland, respiratory tract, and kidney में multiply करेगा. But tell me, if it has to sleep, where does Shah Rukh Khan go to sleep? At his house. His house is Mannat. So from Mannat you will learn mono. Where does it go and sleep? It is going to go and sleep in the monocyte. So repeating this story, how is Shah Rukh Khan working? Megalo. Working में megalo याद आएगा. So salivary, respiratory, kidney, that is SRK, and to sleep goes to Mannat. So for latency, it is going to the monocytes. Now coming back, how is the transmission? So now think logically. It was in your salivary gland, respiratory tract, SRK, salivary, respiratory, kidney. So how is it going to spread? Oral secretions, respiratory secretions, body secretions, that is the method. But a less common method which everyone is aware of is transplacental, right? Mother to child wala method, of course, for CMV we know. And if I'm saying mother to child transmission hota hai, can I say the infections can be congenital and perinatal because mother to child is possible? We all know that congenital cytomegalovirus pe question zyada aata hai. Do you remember the picture that I had shown you in the main videos? And this is the picture of the cytomegalovirus causing congenital infection. And what is that? You're going to draw a cytomegalovirus. So you made the face of this baby over here. So this tiny little baby you have drawn, and around him you have first written the C For his cap, you have written the M, and for his beard, if it's a baby can have a beard, or for his body, you have made a V. What does this tell you? The C tells you, firstly, you'll say the C is alphabet C. So C stands for chorioretinitis. And because these are looking now like ears, they also cause deafness. Next, what is this M going to be? Mental retardation and microcephaly. M is sorted. V will tell me which kind of calcification, parenchymal or cerebral calcification or periventricular calcification. Why am I telling you that? Because the same features are seen in toxoplasmosis also. Toxoplasmosis shows you the same features, but over there we get cerebral calcification. Whereas in cytomegalovirus, the V that you have made will tell you that it shows you periventricular calcification. So repeating once again, what all do we have? The C looks C for chorioretinitis. The C looks like ears, so sensory neural hearing loss. M for microcephaly and M for mental retardation and periventricular calcification. Now coming to, if this did not occur mother to child, what else methodology can be applied? If not through the placenta, then it can also go in the perinatal region. Perinatal region infections usually pass off as asymptomatic and do not trouble much. Same goes if the patient was immunocompetent. Agar immunity theek hai, let's consider an adult or let's consider a grown-up child who has good immunity. In those also, either it will go ahead as asymptomatic or it might show you infectious mononucleosis like symptoms. Now, what was infectious mononucleosis? मैम वो तो एप्स्टीन बार वायरस था. हाँ, तो that same Epstein Barr virus, infectious mononucleosis, glandular fever, those kind of symptoms will be seen if the patient is good immunity. But if the patient has a bad immunity, that is, if the patient is immunocompromised, then CMV, chorioretinitis, is one of the very famous findings that you get over here. CMV chorioretinitis is what is noted. I hope everyone settled with cytomegalovirus. I can go on to the lab diagnosis and the treatment. For the lab diagnosis, I think we all remember it has something known as the owl eye appearance, and that owl eye appearance is based on what? It has intranuclear inclusions. But does cytomegalovirus also have intracytoplasmic inclusions? I have taught you this in your general virology. Yes, it has intranuclear and intracytoplasmic inclusions. That is how the microscopic examination looks like, owl eye appearance. But microscopic is never the final thing. Now you have to isolate the virus. I have taught you in general virology. How do you isolate the virus? What is the cell line? We were using a secondary cell line known as the human fibroblast cell line. And from where will we get the patient sample? The best sample that we get is that of the urine washings. The best sample that we get is of the urine washing. So repeating, virus isolation using which cell line? Human fibroblast cell line. Learn it as it is. Antibody detection can be done, antigen detection can be done, polymerase chain reaction can be done. Honestly telling you, this can be done for every virus on this planet. Antigen, antibody and PCR. So this you just have to copy paste everywhere. But why have I written it specially for this virus over here? Because they ask you which antigen do you detect. And remember, the antigen for cytomegalovirus is the PP65 antigen that you have to know of. Yes, coming finally to the treatment aspect, the drug of choice is going to be ganciclovir, the acyclovir ganciclovir series. And if there is resistance, we go in for foscarnet. So this was about cytomegalovirus, which is human herpes virus 5, and that makes us now go on to human herpes virus 6. So for example, if this is human herpes virus 6, which cells of the body will it go to? It will go to the T cells. Via which receptor? CD46. Everything will come around 6 over here. So yaad rakhna that it is going to go to the T cells via CD46. So human herpes virus 6 goes to the T cells via CD46. It has two variants, that is 6A and 6B. That is also simple. The mode of infection is 6 secretions. So that is how you learn it. This is 6 secretions, right? So the mode of infection also has a 6 in it. And finally, please remember that is the mode of infection usually. But in children, there's a very famous disease which is also called as the 6th disease. And the 6th disease is known as roseola infantum or exanthum subitum. I'll repeat it once again. When we talk about the 6th disease, it is transmitted by 6 secretions or secretions. It is human herpes virus 6, which has 6A and 6B. It is going to enter the T cells via CD46. Has everyone gotten okay with the 6th story over here? Now, when I talk about this 6th disease, what exactly is it? 6th disease or roseola infantum or exanthum subitum, I can see number one, two, three. Three things that you have to know. Number one, I can see that the child is having fever. Very well put up. Yes. Next, I can see that on the palate over here, I can see that there are definitely some spots. What are the spots on the palate known as? They are referred to as the Naga-yama spots. So please remember, Naga-yama spots are seen on the palate. Very famous question. And finally, we are going to have, once the fever will start going away, we will start having these rashes. So rash occurs after fever. You can see these rashes. They are like papules. They are non-itchy and they are pink in color. You can see that very, very well. So this is all a part of the sixth disease or roseola infantum or exanthum subitum. Remember, there is fever, there is Nagayama spots on the palate, and there is a rash that is present. I hope you're not getting confused. There is something called Nagayama, which I am seeing over here in the sixth disease. And very shortly in the RNA viruses, I will teach you something called Nakayama strain. Nakayama strain is something that we use in the vaccine of Japanese encephalitis. So that is totally different. Don't get confused between them. Okay, that is about human herpes virus 6. 7 also tends to show you very, very similar features, so I won't be teaching you anything in detail. Last thing that I have to teach you is human herpes virus 8, and yeah, what do we have in it? Human herpes virus 8 causes 3 diseases. One is a sarcoma, one is a lymphoma, one is a non-cancerous thing. So sarcoma mein to I think everyone knows a vascular tumor called Kaposi's sarcoma, which is most commonly associated with what? It is seen in HIV positive or some kind of immunocompromised people. What is the most common site? It usually occurs in the skin of the lower limbs. Skin of the lower limb is the most common site that you have. What is the second most common site? The second most common site is going to be the lymph node. So these three questions are evergreen questions. Kaposi's sarcoma, which is a vascular tumor, is going to occur in HIV positive patients, most commonly in the skin of the lower limb, and is going to occur other than that in the lymph node. Other than that, guys, what else does it cause? Primary effusion lymphoma, effusions. Patient is going to have pleural effusion, pericardial effusion, peritoneal effusion. बहुत सारा effusions होगा, but that effusion is because the patient has a lymphoma. This information is important only for INI-CET aspirants, that this lymphoma, if you remember from pathology, was CD30 and 38 positive. It is an INI-CET potential question. Coming to the third one, Castleman disease. Castleman disease is a lymph node disease. It is not a cancer. It is a lymph node disease which tends to show you B cell symptoms. Guys, do you remember from medicine what are B cell symptoms? Patient will have fever, night sweats, weight loss, wo sare B cell wale symptoms. Yes, but this is how, again, this information is also important only for NEET and INI, not for FMG students. Castleman disease tends to show you two very important points, and that is, you will see that the cells are going in a round manner like this. Means Castleman disease is definitely going to show you onion skinning of the cells. Onion skinning of the lymphocytes is going to be noted, and suddenly you will realize that okay, the lymphocytes were going round and round and round like this, but suddenly through them a blood vessel has gone. So has that blood vessel become like a stick? The blood vessel has basically become like a stick in the center. So just imagine, now I'm going to draw it once again. For example, if this is a blood vessel, right, and around it you have lymphocyte, which is going round like this. Has it started looking like a lollipop? Yes, there are lymphocytes and there's a blood vessel. That is why you call them lollipop follicles. That's a very famous question that we would expect for NEET and INI students for Castleman disease. Coming back, human herpes virus 8 causes Kaposi's sarcoma, primary effusion lymphoma, and Castleman disease, and we have done all the salient features of each. That wraps up which family? The entire human herpes viridae family is sorted. We were doing the DNA viruses, we were doing the happy mnemonic. When I'm doing the happy mnemonic, so the herpes viridae is done. I'm now starting off with the more common ones, that is the P. I'm starting with the story of the P's, and the P wale mein what we are going to have is the parvoviridae. So now I'll come to parvo, of course. The mnemonic for parvo, I think I've told you a lot of things about parvo, and as soon as I told you parvo, do you remember that story of Aishwarya Rai and Paro that I told you? I told you that Paro was left alone, means it is the only single stranded DNA virus that we have. Paro was also the smallest. She's left alone. She's very, very alone. She's the smallest DNA virus that we have. Apart from that, please note that Paro was left alone. She did not have Devdas. She's single-stranded. She's smallest. So she's crying a lot. Because she was crying a lot, she started having red-colored cheeks. And what is that appearance known as? That is known as the slapped cheek appearance. Now what I want to ask you is, how does this disease spread? So please note, number one, via respiratory route. Next, it can go via a lot of blood transfusions, and it can also go via the placenta, so transplacental. So please remember, transplacenta and blood transfusion usually go together. So parvovirus ka number one mnemonic is that it can definitely go via the placenta and it can go via blood, and secondly, it can go via the respiratory route also. This is one mnemonic for parvovirus. What is the other mnemonic for parvovirus that you have to know? That they ask you, parvovirus attacks which cells of the body? So it is going to attack the red blood cells of the body. It attacks the red blood cell precursors. Which antigen of the red blood cell precursors? It attacks the P antigen of the red blood cell precursors. That's another question. So remember, parvovirus attacks the P antigen of the red blood cell precursors, and that is what you have to know. Coming to the diseases, kya hoga? I just told you, it causes a slapped cheek appearance. You can see that beautiful slapped cheek appearance, not so beautiful, but yeah, slapped cheek appearance can also be seen. And this parvovirus B19 is known as the fifth disease. What did I just teach you as the sixth disease? Sixth disease was what? Human herpes virus 6 was causing the sixth disease, roseola infantum. This is the fifth disease. How have I learnt it? With 5 fingers you put a slap to someone, right? All the 5 fingers should get imprinted. So slap karna hai, so you always have to think of 5 fingers, and that is how you learn it as the fifth disease for slapped cheek appearance, known as erythema infectiosum. So how did you slap? With your 5 fingers. So something will, if you put redness over here, redness will come over here also. So can I say that the rash will occur on the hands and the rash will occur on the feet, as if the child is wearing gloves and socks? So gloves and socks syndrome bhi kehte hain, because there are rashes on the hands and feet also, and the joints are also affected. So there is arthritis and arthralgia. So ye sare rashes on the face, hands, joint involvement ho gaya. But what about the bone marrow? Parvovirus was attacking the red blood cell precursors. It will destroy the red blood cell precursors. It will cause pure red cell aplasia. There will be complete destruction of the red blood cells, or complete destruction of the bone marrow. If you remember, in hemat, in pathology, I have taught you which virus is responsible for aplastic crisis in hemolytic anemias, and that is going to be the parvovirus. Coming to the last one, mother to child. Yes, I told you, P for parvo, P for placental transport ho sakta hai. It causes hydrops fetalis. So these are all, if you learn it in a logical manner, it's easy. Pehle sare rashes ho gaya on the face and the fingers. Then you have arthritis, arthralgia, and aplastic crisis, pure red cell aplasia, and hydrops fetalis. Finally, if I ask you that this P is over, happy mnemonic mein agla P kya tha? The next P is the papova viridis. So if you break papova, out of papova you can get papilloma and polyoma. Are they not sounding the same? We have something called papova, and if you break it, papilloma, polyoma. This is the next family that you will have to learn in the happy mnemonic. So let's move on to papova viridae. What do you have under this polyoma viruses? Let me give you a brief. Polyoma viruses mein MCV. MCV is nothing but Merkel cell virus. So which cancer will it cause? It will cause Merkel cell carcinoma. Very, very simple. Merkel cell virus, Merkel cell carcinoma. What is Merkel cell carcinoma? It is a neuroendocrine tumor that occurs in the skin. I have taught you this in pathology also. SV40. Earlier they used to say SV40 kuch nahi karata, it doesn't cause anything, but now in pathology textbooks it is shown that SV40 is associated with a lung tumor, that is mesothelioma. Coming to JC virus. JC virus causes PMLE. PMLE is progressive multifocal leukoencephalopathy. I hope all of you remember this short form. If not, quickly ek baar dekh lo. Progressive multifocal leukoencephalopathy is what is caused by JC virus. Finally, BK virus. BK virus is going to cause infection in post-kidney transplant patients. In post-transplant patients, it is going to cause an infection. How have I learned it? BK for bad kidney. BK for bad kidney. So you will always remember, acha, this is going to happen or cause an infection in kidney transplant patient. And these are the kind of cells that you see in the urine. Then you see decoy cells in the urine microscopy. When it will cause infection in the kidney mein infection hoga, so something will come out into the urine. Of course, these are the decoy cells. They are very, very big cells with a very, very big nucleus. So decoy kya matlab hota hai? Decoy means trying to copy someone, trying to just chori se, just in a in a very clever manner you're trying to copy someone. So whom is this trying to copy? Socho, such a big cell with such a big nucleus, you'll say, ma'am, this is trying to copy cancer cells. This is trying to mimic malignancy. So that is what I want you to know. Please note, if I start seeing these cells in the urine, I am a pathologist or a microbiologist, and I start seeing these cells in someone's urine, I'll say, oh, this patient has cancer of the kidney or cancer of the urinary bladder. And then I'll call the clinician, does your patient have urinary bladder cancer? And he says, no, my patient just underwent a kidney transplant. And I'll be like, oh, now I get it. It is not a cancer. These are mimicking cancer cells. These are actually the decoy cells. Maybe the patient has been infected with BK virus. These are all the polyoma viruses. Merkel cell carcinoma, SV40 with mesothelioma, JC virus with PMLE, BK virus with bad kidney, that is the decoy cells. Polyomas are done. Now let's come to papilloma. What comes under papilloma? I don't think I need to teach you. Human papilloma virus. This requires 2-3 slides from our side. So let's study. What do we have? We have human papilloma virus. I think all of us know कुछ तो warts वाला cutaneous involvement है. Then there are some warts and cancers which are mucosal. So when I say skin involvement, mucosa involvement, in both of them, there are some low risk things and some high risk things. Means when I'm talking about the skin, जो छोटा वाला numbers है, HPV 1, 2, 3, they have a low risk involvement of the skin. HPV 5 and 8 have a high risk involvement. So please note, there are going to be low risk के साथ क्या होगा? Yeah, 1, 2, 3 के साथ ma'am, there are going to be some skin warts present. But with HPV 5 and 8, I want you to see a condition. With HPV 5 and 8 and especially HPV 5, can you see there are so many warts? Not one verruca, wart matlab verruca, not one verruca, but there are so many warts as if the person has started looking like the branch of a tree. Tree man syndrome. This is epidermodysplasia verruciformis. Difficult name, but thoda sa ek baar pad lo. Epidermo telling me acha skin involvement hai. Epidermodysplasia, something bad is happening. Verruciformis. Verruca matlab lots and lots of warts are present. So many warts are present that the person starts looking like a tree. So remember, HPV 5 and 8 are going to cause so many warts that it is going to result in tree man syndrome. These are all skin related problems. Now let's come to mucosa related problems. Now the low risk ones over here are again chota numbers, 6 and 11. They cause mucosal warts. They are going to cause mucosal warts. What are these known as? These are known as condylomata acuminatum. These are called as condylomata acuminatum, which are going to be caused by human papilloma virus. Don't get confused don't confuse it with condylomata lata, because I hope you remember I had changed the terminology and I told you learn it as condylomatu latu, because that will tell you that 2 secondary syphilis causes this. Condylomatu latu is caused by 2 secondary syphilis. Condylomata lata it was. Here I'm talking about condyloma acuminatum, or you can just learn it as condyloma acuminatum, that is caused by human papilloma virus. It's a wart, so low risk hai, 6 and 11. But look at bigger numbers, 16, 18, 31, 33, 35, especially 16, 18. They cause squamous cell carcinoma. That is why they are high risk, and they cause squamous cell carcinoma where? मैम जहाँ squamous epithelium है. Squamous epithelium is where all? Squamous epithelium is present in the cervix, it is present in the penile region, it is present in the anal canal region, it is present in the oral cavity, it is present in the esophagus. All of these are places where squamous epithelium hota hai. So wherever squamous epithelium is there, human papilloma virus will cause squamous cell carcinoma. Cervical to is definitely something that you have seen. Let me show you a clinical example. Can you see there are warts? Where are the warts present, ma'am? I can see genital warts, matlab mucosa. I can see some vaginal and some penile warts also. So basically what I can see is condyloma acuminatum. Condyloma acuminatum means these are caused by HPV 6 and 11, that is there. So now when HPV, there are two things. Do you know the structure of HPV and how HPV causes infection? So please note, most of the textbooks have given a round wala structure, but for us to identify better, I'm gonna make a diagram like this so that I can bring down all the structures in one end, okay, so that you don't have a problem later on. So dhyan se dekho. First and foremost, I'll put it up as L1 because you need to know that there is L1 capsid that you need to know. L1 capsid kya hota hai? This is the one that you are going to use for vaccine preparation. This is a previous year question. L1 is used for vaccine preparation. Then you have a lot of molecules like E2, E4, E5, E6, E7. These are all the molecules that an HPV has. Please remember, E2 is the controller. Everyone needs a supervisor, na? Who supervises all these proteins? E2 is the supervisor. E4 and E5 are responsible for making a cell called koilocyte. What is a koilocyte? Let me describe it to you. This is a koilocyte. Koilocyte is made by which proteins of human papilloma virus? E4, E5. What is a koilocyte? Let us draw it. You will say, ma'am, koilocyte is like any other pink color cell, but I will realize that the nucleus is a little weird. The nucleus is a very dark, you can hardly see it, a very dark raisin-oid nucleus. That is how you describe it. It has a raisin. Raisin means kishmish. Raisin-oid nucleus. And do you see the cytoplasm? I have not drawn the cytoplasm around it. Iske aaspas pink nahi hai. It is white. There is a perinuclear halo. That is how you describe a koilocyte. So have a look at this. Do you have a very, very dark nucleus over here? Very, very dark raisin, kishmish-like nucleus and a white area around it, perinuclear halo that is present. So raisinoid nucleus with perinuclear halo is what is noted in a koilocyte. Who makes a koilocyte? It is the E4 and E5 proteins. Now what do E6 and E7 do? E6 and E7 are responsible for carcinogenesis. Please note that E6, matlab jo cancers hai, they are the ones causing cancers. E6 is going to switch off P53 gene and E7 is going to switch off the retinoblastoma gene. And let me tell you that P53 gene, I think you know this. P53 gene is known as the policeman and RB gene is known as the governor. So who is more senior? Ma'am, governor is more senior than a policeman. So senior official ke liye senior wala number. The E7 switches off the governor, the RB gene. E6 switches off the policeman, that is P53 gene. So basically I can say carcinogenesis that cancers that are being caused are by inactivation of the tumor suppressor genes. Tumor suppressor genes are P53 and RB genes. These are all the molecules that you had to know for human papilloma virus. Repeating, L1 पे क्या question आया है? Vaccine के लिए. E2 is the controller. E4 and E5 make koilocytes. E6 and E7, they are responsible for cancers. Finally, that you were talking about vaccine over here, finish off the vaccines and finish off this virus. So human papilloma virus, it has a bivalent vaccine which is known by the name of Cervarix, and then there is a quadrivalent vaccine known by the name of, yes, Gardasil. Bivalent matlab it is going to protect against HPV 16 and 18, only the cancer wale. Quadrivalent, of course, cancer though I have to protect, but I want to protect against warts also. So 6 and 11 also it will give protection against. That is what is the difference. So Cervarix is going to be 16, 18 and bivalent obviously, and Gardasil is going to be 4 valencies, so quadrivalent. What is nonavalent? Nonavalent is also known as Gardasil but with 9 strains. It actually protects against nona. Nona means it protects against 9 strains, and these are the vaccines that you have to know. What were we doing, guys? If I proceed further, we were doing the happy mnemonic. What all are we left in the happy mnemonic? Jaldi se revise karo. Herpes viridae family done. Parvovirus done. Polyoma, papilloma, that family is done. What are we left with? You'll say, ma'am, bacteriophage, so you have already taught us in transduction. Then pox virus you didn't teach us. Why should I teach you pox virus? I have taught you everything about pox virus, molluscum, smallpox. I have taught you everything in the general virology section. So jitna molluscum pada tha, that is all that you needed. I am only left with A, that is adenovirus. And I think I have shown you this picture earlier. Adenovirus is going to have, I had told you adenovirus se alien yaad karna, because alien किस में आया था? Alien had come in a space vehicle wala shape. So remember, the space vehicle shape is going to be for adenovirus. And does it have an envelope? No. If you remember, it was a non-enveloped virus. I have taught you this in general virology. So what does it cause? What does adenovirus cause? Number one, it causes hemorrhagic cystitis. Now you will realize it causes a lot of things. Okay, ma'am, that we learn. Problem is you have to learn their numbers. 1 causes what, 2 causes what, 3 causes what. It's a total mess. Try to learn the way I am learning. Adenovirus causes hemorrhagic cystitis. By which strains? By, hemorrhagic. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. The word hemorrhagic has 11 alphabets. So adenovirus 11 and 21 are going to cause this condition. Infant diarrhea. Infant diarrhea is caused by 40, 41. How do I learn it? Because the way you write 40 is the way you write diarrhea, that is potty. So please remember, potty and 40 remain the same. That is why infantile diarrhea or infant diarrhea is caused by adenovirus 40, 41. Coming to the eyes. In the eyes, I can see there is something called epidemic keratoconjunctivitis, which is known as the shipyard eye. And then there is a pharyngoconjunctival fever or swimming pool conjunctivitis. So kya kya hai? Do cheezein. There is a shipyard eye, which is known as epidemic keratoconjunctivitis. So ship mein epidemic ho gaya hai, theek hai? Ship mein epidemic ho gaya hai. Next is going to be swimming pool conjunctivitis. Swimming pool, just remember, the swimming pool water goes not only in your eyes, not only conjunctiva, swimming pool water you also tend to swallow na, to pharyngo bhi hoga. Pharyngoconjunctival fever or swimming pool conjunctivitis. How do you learn this? For the shipyard eye, one of the most common is 8. So 8, if you write it like this, has it started looking like the eyes? Yes. So 8 is the one that is going to be affiliated with shipyard eye or endemic or epidemic keratoconjunctivitis. Whereas swimming pool, ab kaise, have you guys ever learned how to swim? Swimming ka ek rule hota hai na, that first they say you should learn how to float. Then you should learn the basics of swimming. Then you should learn the advanced strokes of swimming. So float karna, to they always say कि 3 days में flotation सीखो. First 3 days they only make you float. Then at the end of 1 week they tell you the basic strokes of swimming. At the end of 2 weeks they tell you the advancements. So 3rd day पे flotation, 7th day पे basic, 14th day पे advanced. Swimming pool conjunctivitis is caused by 3, 7 and 14. Respiratory diseases. Either it can cause upper respiratory diseases, lower respiratory diseases, or respiratory diseases in transplant patients. So upper ke liye chota wala numbers 1, 2, 3. Lower ke liye you have little bigger 3, 7, 21, and transplant TT 30, 30. So transplant 30, 30. Matlab 34, 35 are the numbers. Repeating, for upper respiratory tract 1, 2, 3. For lower respiratory tract it is 3, 7, 21. And for TT transplant 30, 30, 34, 35 are the numbers. Out of this, if you personally ask me exam mein kya aayega? Hemorrhagic cystitis 11, hemorrhagic 11, 11 number aayega. Infant diarrhea, diarrhea 40, 40, 40, 41, simple. These are the two most expected. After that, the two eyes. The shipyard eye looks like 8, so 8. And swimming pool ke liye we have 3, 7, 14. That definitely marks an end to almost everything of the happy mnemonic. But I'm left with the Hepadnaviridae family. What do I mean by that? See, when I talk about hepatitis, I told you it is only hepatitis B virus which belongs to the DNA virus family. All the other hepatitis are going to belong to the RNA family. So, kahan pade? DNA or RNA? Kahin bhi nahi. Let's study hepatitis together so that I can get all the hepatitis viruses under one roof, out of which, remember, hepatitis B is a DNA virus and all the other are RNA viruses. So let's take all the hepatitis viruses together and we'll divide it into a table starting from hepatitis A all the way up till hepatitis E. So A, B, C, D, E, for which I first want to tell you what are the different families. And for their families, there's a very famous mnemonic of learning it. From A, B, C, D, E, they say that, and that's probably the unfortunate fact, but they say that private hospitals favor rich clients. That is how we learn it. So what is private hospitals favor rich clients? P for Picorna viridae, which includes the enterovirus 72. Then we have Hepadna viridae. Then there is Flaviviridae. Hepatitis D actually is something that is always going to depend on hepatitis B. So for R, I have learned it relies on hepatitis B. And C for Calciviridae. So repeating, private hospitals favor rich clients, Picorna viridae, Hepadna viridae, Flaviviridae, relying on hepatitis B, and Calciviridae. In fact, now there's an update which says that hepatitis E is probably not Calciviridae, but is Hepiviridae, which is a separate family that is coming across, which is so easy to learn, Hep E, so Hepiviridae family. Now let's talk about their genome. I think I've already told you that the only one who is belonging to the DNA members is the Hepadna viridae or the hepatitis B. Rest all of them are going to belong to the RNA family. So that is settled for all of them. Now, what is their mode of infection? I suppose this also everyone knows that hepatitis A and E tend to be like cousin brothers. So both of them are going to cause gastroenteritis, food poisoning, gastroenteritis. If something causes food poisoning or gastroenteritis, means the mode of transmission is going to be feco-oral for both of them. Whereas hepatitis B and C are the ones that can get transmitted via parenteral route. So B can get transmitted via the parenteral route. Then we can have C also that is get transmitted via parenteral. But hepatitis B, I think everyone knows there is a sexual transmission. There's a very famous mother-to-child transplacental transmission also that is available. Next, incubation period. While we do not focus too much on it, but the recent INI-CET that was conducted in May had a question on the incubation period of hepatitis A. So let us learn. This is how you learn. So dekho, jo dono gastroenteritis wale the, food poisoning ones, fecal-oral ones, they have a common incubation period of 15 to 50 days, right? 15 to 50. What about the rest? Hepatitis B and D, they always tend to have an association. Na? So inka is 50 to 150. 50 to 150, and then if you combine all of them, means you take the lower of the first and the higher of the second. So 15 to 150, that is going to be the hepatitis C. So repeating, hepatitis A and E, first you do 15 to 50 days. Then for B and D, you go forward, from 50 you go forward 50 to 150 days. Then you take the lowermost and the highermost number. So 15 to 150 becomes hepatitis C. This was the exam question that was recently asked in the INI-CET. Let's move on forward. When I ask you about the envelopes, I think I gave you a mnemonic in the beginning of this series of hepatitis or of this series of general virology, where I gave you a mnemonic part which was for non-enveloped virus. And which hepatitis was coming under those? A and E, the food poisoning ones. The food poisoning ones do not have an envelope, rest all of them have an envelope. Talking about cultivable, the only cultivable one that we have is hepatitis A. That is the only cultivable one. Next, which of them have a vaccine? All of them have a vaccine. In fact, does hepatitis A have a vaccine? B have a vaccine? E have a vaccine? Yes. Hepatitis D vaccine as such doesn't make sense because hepatitis D is dependent indirectly on hepatitis B only. Why is it that hepatitis C doesn't have a vaccine? Now, this is where you will get a question, because hepatitis C follows something known as a phenomena called quasi-species. Hepatitis C follows quasi-species. I can't write down a definition of quasi-species, but I can explain the concept to you. And the concept is, I'll explain it with a very relatable example. All of you are on the different social media platforms, all of you are on Telegram, and you are always complaining that, ma'am, on the group, there are so many people who are unnecessarily, putting up spam messages. But then you say that, ma'am, please block them. So I block one, but then the same message comes from 3-4 more usernames. So even if I block one, can I stop those messages? I can't. Why? Because by the time I block one username, that person has made 10 more usernames. Then I block those 10 usernames, the person has made 20 more usernames. That is quasi-species. Even if you make vaccine, if I make a vaccine for hepatitis C, by that time the hepatitis C will change, it will mutate and make another variant. Then my vaccine won't be applicable or won't be effective. Then I make a vaccine against the new variant, hepatitis C again will mutate. So basically hepatitis C follows something known as quasi-species, and you will never ever be able to protect against all the different changes of hepatitis C, and that is why there is no vaccine. Talking about fulminant, which is the most common in this percentage, the most common cause of fulminant hepatitis, so that is going to be hepatitis D. And if I change the question, which is the most common cause of fulminant hepatitis in pregnancy, then 20 to 25% chances in pregnancy is hepatitis E. So overall pucha, it is going to be hepatitis D, but in pregnancy asked, that is hepatitis E. Talking about the chronicity, please note chronicity, maximum, I can say the maximum potential of having chronicity is with hepatitis C, whereas carrier state, most common carrier state is going to be seen with hepatitis B. So for that matter, I am going to give you a list of the most commons right at the end, or if you want, we can take it up right now. I've got you in the form of, reveal and hide mechanism of what you're going to do. If I ask you, which is the most common cause of sporadic acute hepatitis? Acute hepatitis मतलब वो gastroenteritis वाला. Sporadic acute hepatitis in India, and your answer is going to be hepatitis E. Coming to the next, the most common cause of fulminant viral hepatitis, that I just told you, that see, fulminant viral hepatitis most common is hepatitis D. But if I change the question about fulminant hepatitis in pregnancy, your most common answer will become which one? It will become hepatitis E. Now let's move forward. See, I just told you parenteral route hota hai, blood transfusion hota hai. So if someone asks you most common transfusion related hepatitis, your answer is hepatitis B, because you will realize jitna bhi ye most common wala going forward, I will tell you na, answer will be hepatitis B only. Hang on for it. Most common transfusion associated, hepatitis B. I ask you, which is the most common cause of chronic hepatitis? Most common cause is what I'm asking. Your answer becomes hepatitis B only, because hepatitis B common hi itna hai that the most common cause of chronic hepatitis is hepatitis B. But who has the maximum potential of chronicity? The maximum potential of chronicity is with hepatitis C. That is what you need to watch out for. Dekho, if you look at the overall incidence, hepatitis B is more common. So the most common cause of chronic hepatitis becomes B. But who is more dangerous? That is the more potential of chronicity, that is hepatitis C. I'll again give you a relatable example. So who are the students who top? That is, ma'am, the student who is, consistent, the student who is hardworking, the student who listens to lectures, who practice MCQs, who consistently studies for 1 year or 6 months properly, dedicatedly. That student is going to obviously get a very good rank. So someone who is very following the common rules. But do you also know that one student in the class who's very, very brainy, he can read once and he can, beat any topper, but just that he doesn't study. We all know. So we always know one student who has the maximum potential in him, and that is how you define hepatitis C. Okay. Similarly, if I ask you which of them is the most common cause of carrier state, most common cause ka hamesha answer hoga hepatitis B. B is overall most common. Same way, if I ask you most common cause of viral hepatitis causing cancers, ma'am, most common cause mein again answer is hepatitis B. But who has the maximum association, strongest association with hepatitis C? That is going to be hepatocellular carcinoma. That is going to be hepatitis C. So look at this very carefully. Most common cause of transfusion related, most common cause of chronic hepatitis, most common cause of carrier state, most common cause of virus causing liver cancer. Answer to all of these is hepatitis B, hepatitis B, hepatitis B, hepatitis B. But if I ask you which of these has the maximum potential of chronicity or the strongest association of hepatocellular carcinoma, then your answer becomes hepatitis C. Association, strong, potential, इनसे hepatitis C आएगा. fulminant के लिए hepatitis D, fulminant in pregnancy hepatitis E, and simple acute hepatitis पूछा इंडिया में, that is hepatitis E. So I think all of this is settled. Now you've understood the table, but did you notice other than A, B, C, D, E in medicine, you are also taught something called hepatitis G, although it is not known to cause any human infection, of course. But can it enter the cells? Yes, it can enter the mononuclear cells of the body. Number one, it can enter the mononuclear cells. How would it enter the mononuclear cells, or how will it enter our body? By means of blood transfusion. So hepatitis to nahi karata, but actually can it be protective against something? Yes, it is said to be protective against HIV. So these are some potential questions on hepatitis G that could be asked in the paper. Now, out of these, I know all of you are aware that there are those graphs that you have. You need to do the lab diagnosis of two things, hepatitis A and hepatitis B. So let's begin with the lab diagnosis of hepatitis A virus. So please note that you can see over here in the graph that the patient is going to have jaundice, which you're expecting. But can you also see that there is a graph of fecal HIV? What is fecal? So in the feces sample, in the stool sample, you are going to see hepatitis A virus that is going to be there. And can you see how much prior to the symptoms? From 2 weeks prior to 2 weeks after the symptom onset. So please remember the symptom onset is over here. So from 2 weeks before to 2 weeks after the onset of the symptoms, you are going to see the feces, the fecal sample showing you hepatitis A. So for example, this is the start of jaundice. So 2 weeks before and 2 weeks after the onset of jaundice, you will see this phenomena. This is about the virus. What about the antibodies? In the antibodies, please remember the first one to appear is of course going to be IgM antibody, and the later one, which is 2 to 4 weeks later, that is always going to be IgG. There is no rocket science in it. We already know IgM comes first and IgG comes later. So hepatitis A is a very simple lab diagnosis. What you actually have to dwell in detail of is hepatitis B, and I hope you know hepatitis B is also known as the Dane particle. It is also known as the Dane particle, and it measures 42 nanometers. Now, having said that, please note the genome has certain genes. What genome am I talking about? This was the only DNA virus in this entire family. I know all the DNA viruses are double-stranded DNA viruses. However, for INICET, please remember guys, partially double-stranded DNA, the only DNA virus which is partially double-stranded, that is going to be hepatitis B. So this DNA virus has multiple genes. The P gene is obviously going to code for something P, that is going to be DNA polymerase, number one. Next, we have something called as the S gene. S gene will code for surface antigen. Guys, do we have any other name for the surface antigen? It comes as a very famous name called as the Australia antigen. Then we have something called the core antigen, C for core antigen, and this is going to actually make two antigens. It is going to make HBC antigen, which is known as the core antigen, actual core antigen, and there is also production of HBE antigen, which is said to be the pre-core antigen. So how many antigens have you got? Can you see the final table of antigens being made? There is a surface antigen by the name of HBS, there is a core antigen by the name of HBC, and there is a pre-core antigen by the name of HBE. So what does this X gene do? So what is the concept of HBX? Remember, HBX has been implicated. They put a question mark. They have hypothesized that this is responsible for causing hepatocellular carcinoma. On that note, once you have understood all of these, I can now take you into the lab diagnosis of it. And the lab diagnosis tells me that when I'm talking about hepatitis B, the first one to appear, the very first marker to appear which you see first and foremost is hepatitis B surface antigen. What is the other name of hepatitis B surface antigen? It is Australia antigen. Let me ask you a question from pathology. Is there any stain for the Australia antigen that they ask you in the liver section of pathology? Yes, there is a stain called the Orsin-Shikata stain. Again, something which is important for NEET and INI, not for FMG. Orsin-Shikata stain is what is for Australia antigen. So the first to appear, can I say this is going to tell me the onset of infectivity, that okay, yes, the infection has started. So you can see if I trace it, hepatitis B surface antigen is going to rise and is then going to fall, and I assume कि antigen chala jayega, then the antibody will come. Antibody should have started coming from here, but antibody is not seen. I see a certain gap in between, and then after certain weeks I see that the antibody has started coming up. Anti-HBS comes a little later. So what is this gap gap known as, when the antigen has gone and the antibody has not come, तो वो window बन गया ना, this gap is known as the window period. So you will be like, ma'am, see, diagnosis is very simple. If the patient comes to me in these weeks, the antigen will be positive. If the patient comes to me in these weeks, the antibody will be positive. But if the patient comes to me in this duration, then what is positive? So please note, there is something called as the HBC antigen. Antigen आता है? नहीं. The name of HBC antigen was the core antigen. Core antigen will not be released in the blood. So you will never ever detect it in the blood. The concept is only that, na? It is in the core of the virus. It will never come out into the blood. Antigen will never come out, but antibody to banega na. If it has come in your body, you will make an antibody. Anti-HBC antibody is formed, and can you see that in this duration, who is actually going to help me? Can you see who has covered the window period? Who is covering the window period? IgM type of anti-HBC. So if they ask you what is the marker of the window period, remember anti-HBC of the IgM type is a famous question. Now, did you notice on the top of the graph there is something called HBE and anti-HBE? What does that indicate? So remember HBE, which I am calling as the pre-core antigen, that if you notice, देखो सबसे पहले क्या था? First one was surface antigen. Right after surface antigen, I can see shortly after it, the HBE antigen has come up. So can I say shortly after HBS antigen, HBE comes up? What does HBE tell you? HBE antigen is indicative of high infectivity. So till the time this is coming up in the blood sample of a person, he is very infective. And once it goes away, तो उसका antibody आ जाएगा. Once the antibody has come, means okay, we are in the recovery phase, protection phase, so the infectivity has gone down when the antibody comes. So simple, if I give you a report in which HBE antigen is positive, you will say, oh, hepatitis तो है, but it is very infective. Versus I give you a report in which anti-HBE is going to be positive, anti-HBE antibody is positive, you will say that, oh, now the infectivity has gone down. So now you will always, if I show you a simple graph, we will always learn it with the help of SEMA mnemonic. What is SEMA mnemonic? SEMA mnemonic basically says that you have to focus on, so I'll write SEMA over here. This indicates that whenever you are talking about A for acute hepatitis, the first one to rise is hepatitis B surface antigen. I anyway know that. The next to rise is HBE antigen because S के बाद E आता है. And who's the marker of the window period? IgM type of anti-HBC. So repeating, there is a surface antigen, then there is an HBE antigen, then there is IgM type of anti-HBC. That indicates acute hepatitis. But is this acute hepatitis of low infectivity or high infectivity? You'll say, ma'am, for infectivity, I had to go to HBE antigen. वो है, मतलब this is a case of high infectivity. Let us change the question. Always I have to follow the SEMA mnemonic. तो इसमें क्या होगा? Surface antigen है? Yes, ma'am, surface antigen है. E antigen है? Yes, ma'am, E antigen is also there. E antigen हो गया तो immediately write infective, very infective. Instead of IgM type of anti-HBC, there is IgG type of anti-HBC. So what is the change? IgG type of anti-HBC has come up over here. That means the IgG aa gaya. It is no longer acute hepatitis. It is chronic hepatitis. So please remember, for acute hepatitis, you will learn the mnemonic SEMA. And for chronic hepatitis, you will learn the mnemonic SEG-C. So it's not a very, not a very nice mnemonic, but something that you will learn. So SEMA is SEG-C is how we will learn it. So surface E antigen, IgM, and acute hepatitis. Surface E antigen, IgG, that is chronic hepatitis. So SEMA and SEG-C is how you're going to learn it. Now coming to what if the patient is in recovery? You will say, ma'am, recovery matlab antibody is formed. कोई भी antigen नहीं होगा. No antigen is going to be there. And I will see antibodies are there. Simple rule. If you see all antibody stuff, the patient is going to be in recovery. Very simple. Now let's talk about what is the window period. Window period when now no one is coming positive. Who is the marker of the window period? I know that it is IgM type of anti-HBC. This is also settled. And what is it when we get immunized or vaccinated for HBS or we get vaccinated for hepatitis B? It is always anti-HBS. Always remember कि जो vaccination होता है, that is for surface antigen. So the antibodies that we will form will be anti-HBS. I hope everyone is okay. But was there one one virus which was dependent on hepatitis B always? Yes, and that virus is going to be hepatitis D. Hepatitis D either साथ होगा, that B and D ended up having an infection together, that is a co-infection, or the patient was already having hepatitis B earlier on and now D has come added on top of it, super infection. So simple, either you and your friend enter into the party together, so you are co-entrants, okay, you are co-infection. However, if your friend is already in a party since 1 hour and then you add on to the fun, so you are going over and above him, that is going to be a super infection. So co-infection and super infection are phenomena seen with hepatitis D. I hope you remember this is the one that was causing fulminant hepatitis. Now I want to ask you that if I correlate this information with pathology, what are the different photos that you can see in pathology? Number one, can you see the liver is getting very, very pink? The cells are becoming very, very pink. These are ground glass hepatocytes which are going to be seen with hepatitis B infection. Hepatitis B matlab surface antigen. Surface antigen matlab kaun sa stain? I have already told you it is going to be the Orsin Shikata stain. So ground glass hepatocytes, remember, for hepatitis B. Then asking you one more question from pathology. Where do you see these pink color single bodies, the Councilman bodies? We had studied that Councilman bodies are seen in viral hepatitis. And what exactly viral hepatitis? Which one? Especially seen with hepatitis C. Councilman bodies are seen very commonly with hepatitis C virus. And what kind of bodies are these? That's our first chapter, first question. It is going to be apoptotic bodies. Apoptotic bodies are Councilman bodies. Since I'm talking about hepatitis C, let me also show you one more change of hepatitis C. Can you see a lot of fatty liver changes have happened? Macrovesicular fatty change, big fatty changes are going to occur. Macrovesicular fatty liver hoga, number one. Then can you see some blue spots? That is going to be lymphoid aggregates. These are all the features of the hepatitis C, and you just studied that, okay, Councilman bodies are also going to be noted. So repeating, Councilman bodies are seen with hepatitis C. A lot of fatty liver changes are seen, big fat deposits of macrovesicular, and lymphoid aggregates are also noted. So did you just wonder that, ma'am, hepatitis ka diagnosis to abhi you were doing with blood tests? Suddenly you started showing me so many pictures of biopsy. Do you do a biopsy in a case of hepatitis? When do you do a liver biopsy? Ma'am, we don't do it for acute hepatitis. We do it in cases of chronic hepatitis only when I want to know the staging and the grading of hepatitis. So what is the indication of a liver biopsy in chronic hepatitis? When you want to do the staging and the grading. How do you do staging? How do you do grading? These are things that a pathologist does. So remember, grading, GIN. Grading is done by looking at number one, the inflammation, and number two, the necrosis. Grading is based on inflammation and necrosing. Staging is based on fibrosis. So, ये confusion होगा in the exam. What is necrosis? What is fibrosis? So, remember, grading is based on inflammation and necrosis. Staging is based on fibrosis. Next, I had to show you one chart which I believe I have already shown you in the beginning. So, this becomes very important for medicine also. And one last information which I want to correlate again with pathology is that hepatitis B and C don't only affect hepatic matlab liver, they also have features outside the liver which you studied in medicine a lot. For example, with hepatitis B, is there any vasculitis that comes to your mind? That is polyarthritis nodosa. 30% cases of polyarthritis nodosa are hepatitis B positive. Membranous glomerulonephropathy is hepatitis B positive. So remember, PAN and MGN, these are hepatitis B related. Whereas if you add MPGN, membranoproliferative glomerulonephropathy, you add a hemat disorder called mixed cryoglobulinemia and a skin disorder, very famous, lichen planus. What is it about dermatology? What bodies do you see in lichen planus? Do you see the colloid or the cuvette bodies in lichen planus? Yes. So yaad rakhna, colloid or the cuvette bodies in lichen planus start with a C and they are also associated with hepatitis C. So lichen planus in the skin, MPGN in the kidney, mixed cryoglobulinemia in the hemat, these are all hepatitis C things. Hepatitis B ke saath PAN and MGN. That's about it. And that wraps up all of your hepatitis chapter. Yes. Now, this was a mixture of DNA, RNA, both, right? But now we are going to enter into the world for the next 1 hour. We are going to talk only about RNA viruses. And under RNA viruses, the first one that we will talk about is myxovirus. But before that, we have now successfully finished the entire happy mnemonic of the DNA viruses, including even the not just hepatitis B of DNA, but we have done all the hepatitis which were a part of RNA family also. Now I am only and only going into the RNA viruses. So first let's begin with Myxoviridae family.